Well, hello and welcome to Genomics in the Cloud Book Club, Week 14. I'm your host, KT Picard. In today's meeting, we'll be covering Chapter 13, Assembling Your Own Workspace in Terra. We'll be building on information we learned in Chapter 11, except this time making it your own, or what I like to call terraforming. We'll be presenting, we have a guest speaker presenting this information today. We'll follow our usual format with additional resources and an open discussion at the end. So this week, it's my pleasure to introduce our seventh guest speaker. After the presentation about Terra, you may be wondering how to customize workspaces to fit them to your particular application. Today, we're honored to have someone whose job it actually is to make this technology more accessible. For over 16 years, our speaker has been analyzing multi-scale big data. She is currently Cloud Liaison Engineer at NCBI, the National Center for Biotechnology Information, part of NIH, where she engages with the biomedical community to take advantage of publicly available data sets. She began her career as a marine biologist and has a PhD in zoology from North Carolina State. About 10 years ago, she made the switch to bioinformatics and received a master's in biotechnology and bioinformatics from Johns Hopkins University. Our guest previously worked with Geraldine at the Broad and was a contributor to the data services platform that we now call Terra. I'm pleased that she'll be blending the book material with material that is recently available data sets on NCBI that will make today's meeting highly topical. Please welcome Dr. Adelaide Rhodes.
Thank you. I am talking to you guys today about Chapter 13, but it's going to be a little bit of a different format because I want to actually use Terra for a project that involves the place I work currently, the NCBI. And I'm going to explain that at the end. But first, I thought I would just go through the slides, and this would be an opportunity for anyone to bring up issues they had with the chapter. But honestly, it was so well written, the chapter, I felt like the instructions would be very easy to follow. And we're very familiar with the GITC workspace, but there's so many other kinds of workspaces we can build out there. So I just wanted to kind of go through a case study. All right, so this was in the book. Like, it's pretty easy, to follow the directions and create a new workspace. There's a button that shows up on the front page, and you can name it what you want. You have to link it to a billing project. You can't have a new project, a new Terra project without a new Google project. So you need a billing code attached to it. And we've kind of gone over that in the past. Also in the book, it stepped through each tab in Terra. It went from like the dashboard to the workspace, whittles, and things like that. So that's what this screen is about. It's just talking about making a new method using an existing whittle. So you can go into the doc store or the Broad's method repository and pull in a method that already exists. You don't have to make your new method, and then you can just make adjustments. So that's kind of like the typical thing that I do. It's like I go look for something that's similar to what I'm trying to do, and then I just bring it in and then I make adjustments. And I'm sure you're familiar with, in the book, it goes through all the different settings that you can put into the workflow, such as number of CPUs you need, amount of RAM, whether you want it to be preemptible, all that stuff can be programmed in these whittle files. And then there's a workflow page, and this has changed a little bit, so we'll go look at it. But basically, you can look at your whittles inside the workflow page and figure out like what parts you need to change. And that's kind of what I did. I have a demo where I'm going to show you where I stepped through the whittle to find the files that I need to change, which are in a Docker that are linked to a GitHub. So if I want to recreate this workflow, but then have my adjustments to it, it's really good to understand how to navigate these workspaces. And that could be a little confusing if you're not real familiar with Terra and using Terra. And then the book does a really good job of describing all the different kinds of data tables. They have added some more documentation on the website, but it's sometimes the files, it's easier to like bring a file in and add to a file than it is to actually delete a file. So if Geraldine shows up later, we should ask her about that, because maybe I just don't understand. But I think the book did a really good job of explaining that the Terra, the platform is looking for certain information, such as this first column here where it says entity : you have to have that. So that's something that's unique to Terra, so that it can know what type of thing you're looking for. Because then if you have nested workflows, things that are certain types of entities are recognized as being nested inside other types of entities, if that makes sense. So that's kind of like the most confusing part of this chapter, is understanding how you take a data model, like how you organize your data for your project, and translate it into the organization of your data tables. But once again, if you follow the chapter very closely, it's not too bad. So anyway, here's just an example of how a participant relates to a sample and relates to a sample set, right? So this is a publicly available data set used for testing pipelines, right? Like we always test them on the same individual, because then we can quality control, right? Production and stuff. So this tends to be one that's pulled a lot. It's not the only one, but it's a participant in the 1,000 genome study. And there's a sample, and you can have a large and a small, but this is kind of a simple data model where there's a participant and they took a sample, so it's a 1-to-1, right? But you could see where there's cases where maybe there's multiple samples, for one individual. So that's where you might start getting into sample sets. And this is from a recent blog post. For those of you not reading the Terra blog post, I highly recommend it, because it really gives some more nuance and finesse and builds upon what was already in the book. Like, not a lot has changed since the book, but if you want more real world like case studies and like some breakdowns of how to do this, there's some really good examples. So I pulled this from a blog post, also talking about how sample participant ID relates to sample ID, right? So there's two samples. Maybe you've got a tumor and a normal, right? That's something that you can do with GITK. You can compare a tumor to a normal. So it gives some templates and guidelines for how to like get ready for a mutect. So I put all the links for each slide there. So like later on, if you want to go look it up. Okay, so another thing they went over in the chapter was how to take an existing work data set and then add to it with more data. So one of the recent additions to the federated data sets inside Terra is this 1,000 genomes project high coverage, right? So this is also funded by the NIH through the NHGRI Anvil program. And so they took phase 3 samples and sequenced them to 30x coverage, which kind of brings a lot more precision to finding variants. More coverage is better, right? Because sometimes the 1,000 genomes catalog, which was done almost, 10 years ago, or no, 2015, between 2008 and 2015. So you have to think the tech wasn't as good, the coverage wasn't as good. There might have been a lot more low quality scores. So now they've redone the data set with really high quality scores, but maybe you don't need to rerun everything. Maybe you just want to look at a few individuals that you have a few questions about. Maybe they have a certain set of markers that you're interested in, but you want to make sure they're there. So the book walks through how to bring in that data and make it coalesce with the current data that you have in there. So this is just showing that you have a participant and you can have multiple samples for the same participant, and then you could call it a sample set, or you can have a sample set made out of samples or sample sets made out of participants. It kind of gets wonky in there. So I just recommend reading the chapter about that. And then it just shows you how to copy the data into the workspace. And I thought the most significant thing that it mentioned in there is like, okay, so you're going to upload the metadata for your 1,000 genomes data, and you're going to add it, or the high coverage set, the 1,000 genomes high coverage data set, and you're going to bring it in and be comparing to samples you already have in your data table from the low coverage. Be careful, because the data tables are sensitive to the column names. So if the field names are not done in the Terra way, it's going to not recognize that it's the same data. So for example, if you've got cram here, but then you've got it, or like actually the example they give is GBCF. I think it's all caps in one of the data sets, and there's a lowercase g in this data set. So what you have to do, and it walks you through this, is download the TSV. This is the simplest way to adapt it at this time. There may be more options in the data table view at this point, but I'm, I was just playing around with it this morning. I don't have all the knowledge, but there's information on the help database. You can always write into the forum that you need to just kind of go in, download that TSV, and then rename that column header, and then re-upload it. And then it's gonna still have the old data. So that's the other thing I noticed. It's very similar to how BigQuery works. Instead of replacing data, a lot of times it's just appending data, which is a little bit confusing. So when I was trying to put in a new sample set, I was like, I just want to delete the old sample set before adding the new sample set, because it doesn't have like an easy way to do that. So it kind of appends the data, but that's okay, because you can give case studies like where really I just want to append the data. I don't necessarily want to start all over again with a completely new list of things. And so the example I'm going to give at the end kind of would work on that model anyway. So, but problem is, if the columns are not named the same, it's going to make a bunch of columns that you don't need and you don't want to look at, but you can hide them. So I did find that feature. All right, and then this, the book just kind of goes through the TSV, and then it also warns you that if you have a sample set and you want to upload the files, it's not just one file. There's actually two files per sample set. There's like one that's like membership in the sample set, and then like other attributes of the sample set. So I was playing around a little bit with that, and I can show you what that looks like if you're interested, but it's pretty easy just to download, like go to the GITC tutorial website that we've all had and just try to download sample set and see what happens. It gives you two files, not just one. Okay, and then it went through an example where you were updating the data and adding samples. And like I said, this is going to be similar to what I would be doing with my case study. And then it shows how you have your old data set, the 1000 genome, high coverage. Oh, I guess it was the high coverage was the one that's 2504. And then you added 25 entities, and then you may add one more. And so you can see that's like a typical researcher caseload, right? Like you may get sequencing data for half your cohort very early on, and you want to go ahead and plug it in, but you don't want to have to redo all the work when you bring the next stuff in. So having this ability to just append stuff to existing data sets is really, really helpful. And then input configuration. It walked a little bit through like the this and the workspace dot. That's another Terra kind of centric thing. It's really just saying make a column name that has this name. Like if I want an output from joint genotyping in my data table, right? So what it does is it puts the data back into the data table that you started out with. The one that had the inputs becomes the one that has the outputs. It adds a column called GBCF, right? In the table samples. Does that make sense? So then it also does that for the workspace things. You can go into the workspace. Oh, and this is another screenshot I pulled in from the Terra helpful forum. I think this was written by Ali Hajan. Her stuff's very good. She's very good at documenting this stuff. And this just shows the complexity of bringing in a set of data sets that are linked from Mutect. But I liked it because it gives this little mathematical formula. Since I started out in physics and math, it says it follows the order A to B, means that entity A must be uploaded before entity B. So if you're going to do combined data tables that rely on each other, think about what the dependencies are. So it's the order of operations should be like what depends on what, and for downloading. So, the samples don't mean anything without the participants, right? So load your participants, then load your samples. If you have tumor normal, then after you've loaded samples, then you can put pairs, like which one's tumor, which one's normal. If you want to make sets of participants, cohorts, people from different regions, do your participants and then your participant sets. Okay, and then this is I think the last slide from the generic, stuff. It's a screenshot of the doc store. So this is what happens like when you go to bring in a new workflow. It brings up the doc store, so you can look for things that are WDL, and then there's a button these days. I don't know why it's not showing in here, but I have another screenshot later that shows it, that says just bring in the bringing this workflow right into your workspace in Terra. So that workspace that you may have created, you can automatically bring it in. You give it the name of the workspace, it recognizes it, and then you can bring it your workflow. Okay, so that was what was from the book. So I'm going to pause here and see if anyone had any questions about this chapter before I move on into my case study.
Looking good.
Okay, all right. We got a small group today. That's fine. That's good because this is all new. It's all hot off the presses, guys. So would love to get your feedback. Okay. All right. So now
I have one very quick question.
Sure.
Good question. Maybe I haven't tried this, so maybe this is a stupid question. When you read in the columns, do the columns have to be in the same order? Do you have to order them, or it just goes based on the name?
It used to automatically alphabetize the column, so that would be annoying if you had a very large worksheet and you didn't want it that way. I don't think it forces alphabetization order anymore, but yeah, like it doesn't matter what the order is because every entity that's like every column has to be uniquely named. That's something to remember because you can't have two columns with the same name. So, you know, if it's this dot samples dot GBCF, that's the only column that can have that designation. Yeah. That's a good question. Other questions? Okay. All right. So my case study, changing an existing workflow. So as I mentioned, I'm going to have all the links in the presentation that's handed back to you guys. But a few months ago, well, I guess it's been 8 months now. I went back and looked at the dates. I can't believe this thing has been going on almost a year. But anyway, Broad was one of the first groups to really just jump on the bandwagon of trying to get like data and push it out there. And not only data, they made a Terra workspace. And it was done by Dr. Danny Park's Viral Genomics group at the Broad, which is great because there's research specialty groups that do computational biology. They're not all in the data science platform. I know that's a little confusing, but a specialist group might come up with a method and then they'll hand it over to data science platform and they'll play with it and make it all nice and put it into Terra so everybody can use their workspace. It becomes a citable thing for future publications as well. And there's a lot of great interaction when you're in the data science platform side. That's where I was working with the researchers directly as they're developing these methods. Anyway, they developed a method that was linked into NextStrain, which is a visualization, I think, platform for viral stuff, right? We want to see how closely related all these viral genomes are. And the workspace started out with, they have an assembly tool in there. They have something called symbol_ref-based whittle, which uses best practices to bring in COVID sequence data, assemble it, and then start looking for variants and things like that, whatever you want to do downstream with NextStrain. And the starting point of this workspace, which was written 8 or 9 months ago, so that's important context, was either FASTQ files downloaded from the sequence read archive or a list of SRA accessions that you would then use this tool called FASTQ dump or SAM dump actually to make BAM files to bring in and make unaligned BAM files from SRA files. And so my motivation for updating this workflow is that during the same amount of time, these 8 or 9 months that have gone by, the NCBI has made publicly available data sets in Google buckets directly, right? Like they're still accessible using the SRA toolkit. The way that works is you tune your SRA toolkit to recognize you're in Google. Like if you were just opening up a VM and using, you know, the SRA toolkit, you would first say, I am in Google, because then it would automatically pull the data in from Google because it's, you know, probably in your region, it's faster, things like that. It's a publicly available data set. But the difficulty when you're using Terra, right, is that you're not opening one VM. You might be opening 50 VMs, right? And I don't want to have to go through tuning the SRA toolkit, which actually is a little bit I mean, I'm going to work with the team to find out if there's another way to do this. There's no command line simple way to do it, if I am understanding correctly. So I want to do a little more exploration and figure out if there's an easy way just to make it recognize I'm in Google. But what I could do is point directly at the Google buckets, like as my starting point. So instead of starting with a list of accessions, I can point at the buckets and then I can start my workflow from there. Does that make sense? Like what I'm trying to do?
Yeah, you bet.
Yeah, so that was the first development. And then the second development from the NCBI was the development of new data objects. This is something that hasn't gotten a lot of, you know, outside external outreach done on it yet, but it's something I'm working on this year. It's one of my main job duties, I guess, would be trying to get people to understand about these new data objects. So the thing is, we can pre-process a lot of the data at the NCBI to save researchers time. And what we could do, and we have been doing, is to provide a few more options besides just bringing in fast queues or, you know, BAMs from those fast queues, or making you do the assembly. We're pre-assembling, we're making sets of contigs that were created from the raw reads. So you can bring in the contigs as FASTA files, or they can be read-aligned back to the shared reference. And then that gives you a SAM file as a starting point. So let me just show you. So this is just an example of a bucket. So there's two types of buckets. There's the run bucket and the SRA.src bucket. So the run bucket would be SRA files, things that have already been put into the SRA format, but you don't have to start there. You can start with what people are submitting directly. Not everybody submits FASTQ files, but you can certainly search, like just by doing this, like if there's a certain file format you're looking for and what people have submitted, just go for it. So I'm putting the link in here so you guys can start looking through these buckets and see what kind of data objects are already there. But like I said, we're already creating data objects. Oh, where's my pictures? Hold on. Oh, here we go. So this is the existing workflow, which is you can either import FASTQ data. So you may have private data that's not up in the bucket. So this would be the kind of the workflow you would use, or you want to use the public data. So the SRA ID. So we're getting thousands of sequences getting submitted every day, right? You can imagine the power of not having to do an extra step, you know, like if you're trying to append to your Terra table and keep going, it would be helpful to not have to do the fetch thing, like if you just point at a bucket, right? So my idea one is direct ingress from GCP buckets. Can I rewrite the WDLs to have direct ingress and not have to do the SRA fetch tool or any kind of tool to do it? Just point at the bucket, start working. And then my second idea, and these are just ideas because I haven't had a chance to test it, but I'm going to put out there at the end that I'm looking for help, is pull in we're calling them sequence read archive read aligned formats objects instead of doing the assembly manual. So these are the contig FASTA files or SAM files that you can find now in our stuff. So the challenge is, can I do these 4 things to take the original viral genomics workspace and make it more tuned to NCBI data options? That's really what this is about. So I would like to test how quickly the direct ingress is versus the eFetch option, which is already written into the WDL. I want to see if I can replace the assembly portion with our pre-assembled SARS objects. And then I want to use the notebook that's already in there to parse out like the quality of them. Can I compare the one method to the other? And does it actually, do we lose quality or do we gain quality? Do we lose speed? Do we gain speed? You know, there's lots of things I can test with this workspace once I get it up and running. And then I would also like to build new notebooks to take advantage of BigQuery metadata sets. So I put a link out. The Google Genomics group has provided the metadata that goes with these samples. So you can imagine how powerful that is, because then you can spend your time filtering down your cohorts of interest using the metadata tables before you even start this process, right? So this would come before the bringing in your SRA data. This would be, coming up with an interesting question that you're going to ask with the data, and is getting the samples that are directly relevant to that question instead of everything. But you might want to do everything. So this workspace could be multi-purpose, I guess is what I'm trying to say. So let's see, and then, oops, going backwards. Oh yeah, and then so this is an existing notebook that I thought I might add to it. So we also have a section of NCBI. So not everything is just raw sequence data. We actually get genome submissions and things like that. So there's another section to NCBI that would be helpful. It brings in RefSeq data sets for viruses. So these would be genome assemblies and stuff like that. So you can imagine why that would be important if you can bring in more of those into your workspace, because then that would give you some more variability. Like you could align, if you are using the part where you align to, you could test like which reference seems to be working better, or can you make a pan reference, things like that. That would be cool to play around with too in such a virus workspace that harnesses the power of the NCBI data. So anywho, so before I move over to what Terra looks like today, after I've been playing around with all this stuff, does anyone have any questions about this project?
Are there plans to make additional data sets available on Google Buckets, or is this a first?
The bucket, it's been there for a little while, right? Like that's why I'm saying I'm trying to do more outreach about it, because this is not going to the cloud is new for NCBI. And so as you can imagine, things that work really well in on-prem don't always work well in the cloud, right? Like matching up the metadata or matching up the folder structure, things like that. Because like, for example, Google objects are flat versus hierarchical, like whereas your folder structure in a regular on-prem machine is hierarchical, meaning you actually are going down into folders, whereas in like a GCP bucket, it's flat. It's like, it's just another location. It's just a single line that gives you a different location. So everything's kind of treated equally. So there's been some technical behind the scenes work to get that to work. But I did talk to the engineers this morning, and they don't want to make any promises. But the idea is that it's just like the NCBI. When you go to the NCBI and somebody submitted a bunch of new sequences last week, and like, so if last week you searched and they weren't there, but the researcher put a bunch of new stuff in, after like a certain number of days and going through production and quality control and all that stuff, then that would show up, right? So a search in NCBI last week would be different than a search on the NCBI web GUI this week, right? Same concept. Cloud is going to be the same. Cloud is the data that that eventually our web GUI will be pulling from. It will be pulling from these cloud data sets. So these cloud data sets will be as up to date as what you're seeing when you were progressing through doing searches over time on the NCBI. Does that answer the question, KT?
Absolutely. Thank you. That's great.
Yeah. But please forgive us. It's all new. And that's why we're going out into the world and doing outreach, and we want feedback, because these SARFs, like for example, these sequentially aligned read formats, like that's just our guess, like what researchers want. You guys might want a different starting point. So we have been adding things like VCF files. Like you may not even have to go through all GITC for some of these really well annotated and smaller genomes like a virus. So that's exciting too, because then your jumping off point is all the post visualization stuff that you want to do with that GITC stuff. I mean, we're never going to not need the regular pipelines, but we're trying to provide different launch points for different purposes, right? So clinicians maybe care more about the VCFs. They're not as necessarily interested in the raw data. Does that make sense? So that's kind of the concept that they're working on. Okay, so now I'm going to start jumping around through Terra. So this was the blog post that caught my eye. Wow, last April. And move my computer here. Sorry, I'm like double screening here. And this kind of, I was like, oh, cool. Like there's a new workspace that's just viral genomics, and like I said, the starting point is SRA. And they also linked it into these downstream tools called Augur and NextStrain for phylogenetics. So this really caught my eye and it stayed in my brain. And then I wasn't working for NCBI yet. I just started working there last year after this was all developed. And I was like, found out about all these new cool data sources. I was like, oh, that would be a good one to take. So here's the original workspace. I'm just going to walk you through about what is good about this workspace for those of you making workspaces that are going to be in the public view. This was linked out from that blog post, so it's important to have all the information there ready to go for researchers who want to do this stuff. So have we talked about this yet in book club?
The what's in the workspace itself?
Yeah.
Other than no, not directly. The book walks through, you know, copying from the previous example, but that's it.
Okay, great. So good, because I don't want to bore you guys if we've already covered this a million times. There's a dashboard. So dashboard best practices. So you want to describe what it is. You want to say a little bit about the data, like where's the data from. You might want to say if authorization protocols are needed for this data. And then it will tell you, give you usually some guides like how to use this workspace. And then this is a great little schematic. So that schematic you saw in my PowerPoint slide came from this workspace that says, oh, these are the steps that are possible with the whittle. So this is a whittle, this is a whittle. So each of the little green headers is the name of a whittle. And so they named their whittles the same as their diagram. Believe it or not, not everybody is that considerate, you know what I mean? Like be consistent in your naming, I guess is what I liked about this workspace also. And then they show, oh, there's, you're going to need to run the Kraken and the viral assembly if you want to use the Jupyter notebook that's in here. That's helpful too. So I liked how they did that. And then it says the ultimate output goes into this thing called next string, which I'm not real familiar with, but sounds really cool. And then they've gone for each of these workflows, like getting the import from SRA data, they give you a version and they tell you what it does, and it tells you what the expected input is, right? And what the format is, and then what the outputs are. So this is like getting metadata from the SRA runs, right? We want to know the accession, library ID, run date, and all that. And then it does that for each of the workflows that are included in this workspace. So that is awesome and saves a lot of time. And then it gives you what's required for input. So it preloads a bunch of databases. Like you don't have to do this again because these are in public workspaces and they've already put the full links for you, right? See pathogen public DBS, right? So the only reason you might want to change this in your workflows is that you have a different Kraken database you're using, or you have, you want to use Silva database or Green Genes or something else other than the one that Kraken provides. And then it gives you output. So for each workflow, it tells you the inputs, any kind of workspace parameters you need to set, and expected output. So I like that a lot. And then this is about tree building. It really is crazy, but then it gives you some hint, like if you're willing to put in the work and do this workspace, these are the outputs you can get. So this workflow this workspace only has 68 samples in it at the moment. So you can imagine if I plugged in like 100,000 samples, how much cooler this map might start getting, right? So that's kind of the point. And yeah, you don't have to use all the pieces of a workspace, but the part that I'm just trying to fiddle with, like I said, are here and here. Like, can I replace import SRA data with my method? And then can I replace viral assembly? So then I had to understand a little bit more about the workflows. Oops. Put that over here. So what I did is I went into the workflows and I said, I need to change the fetch SRA to BAM. So if I click on that, right, it gives me a source, right? So I want to know what's in this workflow. How are they doing this? So the first place I would look is in the workflow of interest, but then I open up the script and I see, oh, the first step is they pull in another whittle that I can't see, right? And then they're going to do this stuff with it. So that was kind of like crazy. I was like, oh, I just wanted to see the script. Because sometimes in here, it's like literally like the bash script that it runs. I was like, okay, I'm going to have to do a little more digging here. So I went over to the GitHub in the doc store. So I opened up the doc store, and then it gave me the source code. Like, where's the source code? Because that NCBI Whittle tool was kind of opaque to me. And then if I went into the doc store and I found it, I just kind of jumped ahead, but you guys can get there from the doc store. It's in a publicly maintained pipeline data set that feeds into the workspace. So somebody's been doing a very good job with documentation, and they've been keeping the GitHub to doc store to Terra going very seamlessly, right? So anytime an engineer is changing the GitHub, it's going to hook into the doc store, it's going to hook into Terra. That's why versioning is important, because you want to make sure if you're trying to reproduce a workflow from a paper, you go pick the same version that was used in the paper if you want to see exactly the same outputs, right? But here is where what I was wanting. I wanted to find what's the script. And so these are tools that are found in NCBI. So these eSearch, eFetch, if you see that stuff, that's an NCBI tool. That is a program that's in the Docker that you run. And another tool that you run is this thing called SAM dump. So this means like I have an accession number and I want a BAM file from that accession number. This is the script that somebody wrote, this little bash script that's inside the NCBI Tools Whittle, which is imported into the Whittle I found in the workspace. So this was like a little bit of an unusual case study because it was 2 steps of Whittles to dig down to get to the bash script that I want to change, right? So really, I would replace this with, I like how it pulls out metadata. I still want that. So the first step wouldn't be eFetch. It would be, go get these, go point at this bucket in the Google Cloud. See, so this is the part that I would be changing, just that one line in order to test my idea for idea 1, right? So let me just show you something else about the doc store. So like I said, there's versions. So there's info here. So this gives me like the version. There is, how you can use it if you want to run it directly without, you don't need Terra to run it. This gives you the what's been happening in the past, like what's changed between versions, right? And so you can see it. This has recently been updated. It's been modified just yesterday, right? So that's what I mean about the people who conceptualize this project. The Terra project is never really just about the Terra workspace. You want to have a very good GitHub repo, kind of relationship that's underlying your workflows so that people get the latest, greatest, or they can go back in time. So I can go back to all these older versions if I want to. And then this is kind of where I went in. I was trying to find like that, what is this task? And then I was trying to find the origin. And so, this is what that is called inside the Docker. And I was like, oh, I just want the script. And so then I found it, like I said. So anywho, that is kind of like what's my process. So I just wanted to show people that. And then this is what I'm talking about over here with Terra. If I click on this link here, you can export to, well, there's more platforms in there. There's Anvil, there's Terra, there's DNA Nexus, DNA Stack, and now another NIH federated data platform is going to be Biodata Catalyst, right? So you can take this and put it into a workspace that exists. So my drop-down box here is the workspaces that I have, right? And I love this disclaimer, DocStore cannot guarantee that this works. It isn't going to change, but that's fair enough. So like I could do that. I already have that workflow in there, but yeah. So anywho, that's kind of how, like when you're creating a workspace, like you might have just gone to the doc store and found that and then, had a brand new workspace, but I'm trying to do it. So here you can see now I've got the workspace twice, but they're identical, right? So it doesn't matter which one I use except how it's tuned. So how is it tuned, right? So you've got inputs. So maybe this is the newer one. Ooh, I shouldn't have done this. So I want to go back to the original. Sorry, I kind of messed up things. But anyway, here's the inputs. So what is it expecting for inputs? So it's going to look for a list of accession numbers. And then outputs, this might be where you have different data tables and you name the attribute by this, like this new name of a data table, and then what you want to call that output, right? This is where you would do that. I haven't had a chance to test it, but I'm pretty sure that's where you do it. And then, yeah, and then so you can auto load that. So like here, if I go, I kind of messed it up because I added the workflow twice, but here, oh, here, it's not doing that. So here you see this. This is going to be a column called library ID. This is going to be a column called biosample accession. Yeah. All right, and it's going to do it on each SRA number or SRR reference is going to pull up a machine and start running this program called eFetch. In my version of the workspace, it doesn't do any of that. It just says this is the location. So it doesn't have to start a machine and go off and spin anything up with Cromwell or do anything. It's just going to say starting point is here. All right, so I was playing around with it. This is only an error because I stopped it, but I was just kind of curious like what it looks like. So here's another thing here. So I ran the workflow, the one I was just showing you. I only picked 7 samples, so let me show you that. So when you're testing, you may not want to do everything, but oh, here we go. Like I can select data. So fetch SRA to BAM. So if I could have multiple versions of that workflow loaded in my workspace. I don't have to just have fetch SRA to BAM latest. I could have 2.18, 2.1.7. So what kind of entity? So this gets to be what the instructions on the dashboard help you with, because sometimes you're going to want to have the input be a sample is expecting a list of things, or a sample set is expecting a group of things. So sample, and then you can select data, and then you can either select all 68 samples, or you can just get specific rows. So that's how I did the little sub sample set. I just went and said, I want to run these. So you can see here that some things don't have outputs. So this .aligned_BAM is one of the outputs that was in the JSON. Some of them didn't successfully assemble, right? Like, so there are blanks. So this isn't combined input output table. So one of my tests is that I just took out all the outputs and I only had my inputs, the first 2 columns, and I started running it. And I just didn't have time to finish it before here because I wanted the notebook to work. So I had to put this data back in. But these are all outputs that were done here. If you hover over it, you can see they're in a cloud bucket that doesn't belong to me. So this workspace, when they ran it, they made all the data publicly available. So I don't actually have to rerun David Park's workflows. All the data that they ran on those 68 samples is already in their data table and pointed to a public space. So if I wanted to go into what David Park already did and just pull the BAMs out to compare to the BAMs that I would download from the NCBI, this might be a place I would go. And then, like I said, there's like cool little things here. You can hide, columns. So I could just have this and, just have like that. Like I could just have three or 4 columns that I've selected. So you can hide the columns. They're still there in the underlying data table, but that might be helpful too when you're trying to design the next step of the workflow. Okay. So I can choose specific rows. And when I do that, it lets me like run the, sorry, there's like just a little error here because I doubled up on the workspace. I did something and I have too many screens open. Sorry, right now. Don't worry about it. But anyway, yeah, so you can select and sub-select. So the thing is, what I would like to do is have it be automated so that it would read my data table. And so for anything that's already been successfully run, that it would ignore it, or things that were unsuccessfully run and we didn't have a good output, ignore those too. I would like to be pointing at the GCP bucket or using the BigQuery to filter down my new SRR data sets to bring in and not waste time rerunning the analysis that I already did. So this is going to be a lot of work because I'm going to have to figure out how to preserve the links that were created in the data table for my original runs and not right over there. So as long as the SRR numbers are new, that should work. So like I could bring in new data sets and I could call them sample set on this day at this time, run it, and it would only run the new sample. So that's the power of this being able to select a subset from the data table. That makes sense. And I could even have like an index in the data table possibly that has the date. And then I could be like, I want to rerun everything from July, or like things changed in New York City, we think. So I'm going to go just grab the samples. I'll get the SRR list from the metadata in BigQuery and just only run things in New York City since this month, just to see if I'm detecting the new variant type, right? Those are the kinds of things that people who want to use this website or this workspace on Terra are probably going to want to do, right? So how do I get there from here? Not sure yet, but I know there's a lot of cool work. And so my last slide is just going to be like, help. If anybody wants to help with this project or conceptualize it, hey, it's open source. We all know Terra now. I could really use a hand. But just another little thing I want to show you guys under the hood is like when you click on the job history, it'll take you to your console view. Right, so the console view is always available to you. Just because you're on Terra doesn't mean you can't use Google the regular way. Like you can be inside the console and just be making buckets, making VMs and things like that. But I was just kind of curious, how this workflow was outputting data, or like the workflow. So all of this stuff is happening. I'm not going to need any of this if I can replace it. So that's kind of like why I want to do it, except the metadata. So the metadata part does spit out things like the geographic location of the sample. So this is the metadata, the out geographic location, the library, the model, the platform, the run date. But if I use BigQuery, that stuff's all in there. So my idea is to make a Jupyter notebook that could harness the power of BigQuery. So this is no longer running inside the WDL. Anytime you can avoid having to run a huge workflow, like if I put in 10,000 SRR accession numbers, you can imagine that's 10,000 instances of orchestration that Cromwell has to do, and there's hog rates and all this other stuff. No. I could just go to the data table and download this information and combine it back, right? So that's another idea here. So I just want to quickly show you, last thing I'm going to show you is notebooks. So like I said, I want to use notebooks that have already been built for COVID data to kind of speed up our discovery. And so this was the one I was talking about. That is a notebook that brings in the RefSeq genome data sets. So it's a different part of NCBI than the than the SRA. So SRA archive is sequence read archive. It means the stuff that researchers get and send to us raw, right? And then the metadata is contained in bioprojects mainly. So that's where you would go get the metadata. But gene data sets is a different location. So this is the original version of the workspace. So my version of the workspace, I just went to the binder site that NCBI provides for that notebook. So you can go play with that notebook, and it's live on Binder. For those of you who are not familiar with it, Binder is an open source way to serve up your notebooks from your GitHub directly. And I loaded it into the notebook place for notebooks inside Terra, and then I want to run it. But it shouldn't have any conflicts because it's not relying on any of the data tables yet. But I just want to give you guys a clue here for something that we didn't talk about. I'm not sure how much is covered in the book, but there's a complete set of API calls. So this is an NCBI API call. So that just means talking internet to internet, I think. What does API stand for? Does anyone know the definition? Geraldine, you're here. Do you know what the definition is?
Isn't it like application programming interface?
Right. Application programming interfaces. So it's the actual call when I click on a button that goes across the internet and comes back with information, right? Is that the way to describe an API?
It's more like generically, it's like sorry, caught me off guard, but generically it's like an interface, an agreed upon basically way that you can ask a question programmatically of a system,
Right.
and it will give you a response in a in a standardized way, right? If anybody else wants to jump in.
Thank you. So I just want to mention that in case we haven't talked about it. But NCBI has a few of these. We don't have a lot, but we would like to build more to just bring in the data reports. Like once it's written, this API, it knows what to do. It goes, gets the certain parameters that you want, and then you can like load data and like look at metadata. So here we are, we're like looking at, these are not just COVID, there are other viruses too. And then it gives you some quality, you know, details in here and stuff. So you can imagine that this is a different part of NCBI than SRA, like I said, but it could be, you know, you can use these API calls or the metadata search, or you could use BigQuery. There's so many things we could do with notebooks in this workspace to make the NCBI data a little easier to kind of pull out and start running highly parallel, you know, questions on. So that's kind of where I'm at. That's as far as I've gotten today. I would be excited to hear your ideas and suggestions. We can make it a little class project if anyone wants to join me. And I talk about Terra all the time at NCBI, and they really want me to have a demo to go to leadership about why this should be something that we should be doing, right? Like, and I really want to show them that it's going to speed up the time between, you know, data being entered into the NCBI by COVID researchers and actually outputting useful, you know, discoveries and insights. You know, there's so many ways to do this, but I really feel like Terra could be part of the picture if I could build this thing. Anywho, that's it. That was my spiel and my case study.
Adelaide, thank you very much. Very interesting. I learned a lot just by watching the whole thing today. It was a whirlwind, but I really appreciate the additional insights that you have and using this new data in SRA. I had a question for you around way of working. So in a coding environment with GitHub, typically you would make changes and then, you know, make requests, and those would be, you know, incorporated. For example, your first WDL had an import of another WDL, and when you walk to that second WDL, you'd be changing the NCBI calls, the search and what have you, to the actual Google buckets themselves. So is it a same kind of workflow where you make, you know, you bring down the WDL file, you make the changes locally, and then you post them back into GitHub? Is that typically how people collaborate in WDL when they're working on projects like this?
I mean, yeah, like, I mean, it's production level, yeah, definitely. Like, they're definitely using a repo of some type. They're not always public, where any alteration is, you know, you have a development phase, like where you're testing the code, but then you have the production phase where it actually gets pushed out, and then it's part of the, you know, the new version after it's been tested. I don't know, Geraldine might have more to say about that, since she does this a little bit more than I do. I usually work, I'm a team of one, so I have to volunteer to do my own versioning on things, but I'm trying to make it a practice, like to always do it that way. Yeah.
Yeah, I mean, versioning workflows is definitely super important to be able to, for if only for the reproducibility part of it, and for collaboration to be able to provide. I think one of the things that we're still kind of working on is versioning configurations, because right now, I mean, you can import as you did the same workflow multiple times, right, and then save the configurations differently, but there's not a thing that allows you to share. Well, there is technically in the internal methods repository. There's a way to share a configuration of a workflow, but that wasn't extended to Terra's way of importing workflows. And for example, I think one of the things that would be really neat is if we could version configurations on top of the workflow itself, like have something, and we have the basis of that in the system. I think making that accessible would be really helpful to be able to more easily compare also like what happens if I use different configurations of the same workflow, different input parameters and stuff like that. Right now, the system doesn't really do anything explicit, and you can do it, but you have to do a bunch of setting up yourself. So I think that would be a really neat thing to improve. And also, like, you know, in DocStore, there's the JSON inputs file that's when you set up a workflow in DocStore, you can provide an input file that gives essentially an example configuration or reference configuration of how to run the workflow, how to test it and whatnot. And unfortunately, right now, when you import the workflow, there's not a way to automatically import that inputs JSON too. And I think that's one of the things that would be really neat. Because right now, what you have to do is like, you have an easy one-click import the workflow to Terra, but then you have to go and download the JSON and then re-upload it, which seems a little bit unfortunate. So I think that's one of those things that should be an improvement that we can get done. And feel free to request things like that in the Terra forum. There's a feature request section. You can also vote on other people's feature requests. And I really encourage you all, if you're interested in some of these improvements, to go and make your voices heard, because the product managers do pay attention to that. And you know, the squeaky wheel gets the grease. So if a lot of people request something, it allows them to justify the development time to make some of these improvements, because you can imagine many different things that could be done to further kind of improve the usability and make it like super powered, but have to prioritize somehow. And one of the ways that the usability improvements get prioritized is based on how many people express a need for it and express that this would make a difference in their work. So make your voices heard, and hopefully we can make those things happen.
Yeah, I mean, I imagine one way you could do that is you could save your JSONs, like when you change stuff. Like I think I was in the habit of doing that for a little while, and then I forgot about that. Yeah, but yeah.
Yeah, and you can like independently put them in GitHub or something like that. But it'd be so nice. The system already does so much for you to manage things. Like that would be that is something that is possible to build into the system. It just needs a little bit of work to do it. A little bit. It's always easy to say it's a little bit if it's a different team that's going to do the work.
Well, I'd like to thank everyone for joining us today. Next week is our last chapter. We'll welcome back a previous presenter, Mathieu. He's with us today, and he'll be presenting a chapter on reproducibility. Very excited about that, and I'll jump to it here at the end for our last chapter in the Genomics and the Cloud Book Club. So Mathieu, looking forward to your presentation next week to kind of take us through the last steps of making it all so that we can do it again, and looking forward to your presentation.
Yeah, exciting finale.
Very good. Okay, everyone, thanks for joining us. Adelaide, thank you so much for your presentation today. Really appreciate the efforts, and looking forward to seeing everyone next week on the next version of the Genomics in the Cloud Book Club. Thanks, everyone.